Hi, welcome to this tutorial on how to use the Adaptive Evolution database. Today I will be talking about how to use the TAED search functionality. To begin, starting from the TAED introduction page, please select TAED search. From this page, it is possible to query the database in three different ways. The first way is to search for a specific GI number, for example, 35136, which encodes a human version of ornithine decarboxylase. When I select Search TAED, it finds the associated protein family and all of the relevant information pertaining to that family. It is also possible to search for a specific species name, for example, Homo sapiens. When I select Search TAED this time, it will generate a list of all of the protein families that contain Homo sapiens. Specifically, here I have searched to display only protein families with a DNDS ratio greater than 1, meaning that at least one lineage within the protein family was found to contain a DNDS ratio greater than 1. I could display all protein families that do contain human by simply selecting the other radial button. Lastly, it is possible to query the database using a specific gene name or gene type. For example, toll-like receptor. When I select Search TAED this time, it will bring up all protein families whose name is related to toll-like receptor. Furthermore, it is possible to refine our search by limiting and putting a max on the number of taxa that are within the gene family. For example, a minimum number of taxa of 50 and a maximum number of 100. Additionally, it is possible to further refine your search by also including a specific species. Here we see that this result found 5 different gene families. To understand the output for TAED, simply look at the different column headings. Here we see the protein family, and we see the individual protein family names. Under alignment, we can view the multiple sequence alignment for each TAED gene family. By selecting this button here, we're able to enter into the TAED protein alignment viewer. By scrolling across the top, we can view individual columns within the alignment, and scrolling up and down, we can view the individual taxa within the protein family. This button here allows us to download this data as a phylum interleaved format. We are also able to view the phylogenetic tree for this gene family by selecting this button here, which opens up the TAED tree viewer. More information on how to use the TAED tree viewer and its functionality will be discussed in a later tutorial. It is also possible to see how this protein family relates to the chordate species tree as well as other closely related gene families in the OneZoom TAED tree viewer by selecting this button here. The TAED OneZoom will be discussed at a later tutorial as well. It is also possible to download the trees in both NHX and NUIC format using this button for NHX and this button for NUIC. Additionally, it is possible to view the reconciled gene species tree reconciliation tree in the TAED tree viewer. This tree contains information regarding duplication and speciation events. In this column, we see the TAED selection results. The check mark at the front indicates that a lineage with putative positive selection was detected. By selecting the plus sign, we're able to see the selection results. Here we see that this TAED gene family was subdivided into 5 different subtrees. which were used to facilitate the DNDS calculations. The first row, p-value, has bolded and unbolded characters. When the p-value is in bold, this indicates that the free ratios was supported, and therefore the DNDS value that is reported belongs to the free ratio model from PAML. When the p-value was unsupported, it is left unbolded, and the single DNDS ratio is used. To view the actual PAML results for each individual subtree, it is possible to select the file as indicated for each subtree. Additionally, it is possible to view the ancestral states for each subtree by selecting this link here. Here in the column for PDB, when we select the plus sign, we see 4 different buttons appear. Each of these buttons represent a lineage found within the protein family, mapped to the corresponding chordate species tree that had a DNDS value greater than 1. For example, this button here for Lineage-specific to Insectivora showed elevated rates of evolution. When we select this link, it takes us to the TAED protein viewer. Here we can see the associated non-synonymous and synonymous substitutions mapped to a PDB entry. Positions marked in red are indicative of non-synonymous substitutions. In the top left-hand corner, we can see at position 22, there was a change from proline to alanine. Positions marked in black, or dark black or dark gray, are synonymous substitutions. Here at position 782, we see a non-synonymous change that had occurred. Mapping between the ancestral state and a current PDB structure can be seen in this top row right here. An alignment between the ancestral state and the descendant state can also be viewed as well. The DNDS value for this lineage can be seen in the top right-hand corner. The next column, KEG, when we select the plus sign, shows us the individual KEG pathways associated with the given protein family. When we select a pathway, for example, toll-like receptor signaling pathway, this brings up the information in the KEG database relating to this pathway, which we can further explore. As we see here, the toll-like receptor 7 is involved in the human immune response system and involved in recognizing single-stranded RNA from virus. Lastly, the column summary contains summary information about branches with DNDS greater than 1 that have been scrutinized for DS saturation. The next way to search the TAED database is to scroll back to the top and select KEG Pathway Search. Here, it is possible to search TAED gene families based on KEG pathways. By selecting the button See List of KEG Pathways, a list of all of the KEG pathways found within TAED is generated. Selecting one of these links, for example, AMPK signaling pathway, will show all of the related protein families that map to this KEG pathway. It is additionally possible to refine your results once again by searching for all protein families and by giving a maximum and a minimum number of taxa. Next, it is additionally possible to search through all of TAED gene families based on an alphabetical search of gene names. By selecting alphabetical by name, an index of protein gene families is also generated. Here we can select an individual letter, for example, S, and we see all of the TAED gene families with whose gene name begin with an S. The next way to search the database is using the TAED BLAST. We can get to the BLAST using this link right here. Here we are able to blast the TAED database for specific protein fragments or sequences and determine what protein family they belong to. First, we can give our search a name. I will use the name plasminogen. Next, we are able to enter in a sequence manually, or we can upload a FASTA formatted file. Up to 5 sequences may be added to this file. Next, we select our output options. It can either be in alignment format or tabular. I've selected tabular. Lastly, we can give a max E value and the max number of hits. Here I will use the default values. Next, I select BLAST. The TAED database is blasting my sequence against all sequences found within the database. This may take several minutes depending on the length of your sequence. Previous BLAST results will be shown below. Once your BLAST has been submitted and is finished, select Check the Status. Here we see that my plasminogen is finished. I can see the results by selecting Show. Furthermore, I can download the BLAST results using the download link here. In the BLAST results, it is possible to see further information by selecting the BLAST hit. Here we see the protein family relating to plasminogen activator with positive selection being found along at least one lineage within that gene family. Thank you for watching this tutorial. This tutorial has been about ways to search the TAED database. Please watch other tutorials to understand how the rest of the database functions and how to best utilize it. Thank you.